cancers, what are the three major cancer associated um, death that you see common, the top three in male and female. In the male will be prostate, colon rectal, lungs, prostate, and colon rectal. In female, similar, lung, breast, and colon rectal. Um, and we talk about a little bit of nomenclature, the difference between benign versus malignant, and just looking at the name. If it's in, usually ends in oma is benign, but if it has sarcoma or carcinoma, that's malignant. Now, sarcoma and carcinoma is will tell you whether the tumor or the cancer cells from epithelial origin. If it's epithelial, it's usually carcinoma. If it's more of mesenchymal origin, then it will be sarcoma, right? If it's glandular, then it's adeno, and a lot of glandular, it's epithelial origin, then it would be adenocarcinoma, for example. An important example of an exception to the OMA that is not benign but more malignant is malignant melanoma. Okay. And we start talking about the root causes of all cancer. Of course, it is DNA damage, but cancer is also multifactorial. It's usually involve both environmental and genetic component. And the genetic component could be nitrogenous compound, radiations, definitely a lot of radiation, carcinogen, just a general term, anything that can elicit, can stimulate to divide uncontrollably. We call it carcinogen. One of the famous called Ames test. Ames test shows that if you give these cells any kinds of compound and expose it to time at a high concentration, then it can cause a mutation. So it causes carcinogen. So anything that can causes a DNA damage, causes cells to divide uncontrollably, it is a carcinogen, right? And we talk about the six hallmarks of cancers in general, and that is cancer cell has this, they make their own growth factor, so they're self-sufficient in that way. They able to evade apoptosis, programs that tell them to die, but they don't. They're also anti-growth signal, tell them to stop dividing, but they still continue to divide. Cancer that are move on to the original site, we call it metastatic cancer, oftentimes make enzyme that can degrade the stroma, and tumors that are usually big inside tend to have new development of blood vessel called angiogenesis. In my previous lab, macrophage was shown to be important in the development of angiogenesis. And if you and it turns out that CSF1 is colony stimulating factor 1, what we're going to look at later, but it's usually a growth factor that is stimulated the hematocytoblast goes into the lineage of mononuclear phagocytic cells, or in other words, monocyte. And the monocyte would then, when they leave the blood, they become macrophage. And tumor are known to put out, some tumor like breast cancer can put out CSF1 to recruit macrophage to do what? To involve in angiogenesis. And in this angiogenesis require another growth factor for angiogenesis is what is cell. It's called VEGF, for vascular endothelial growth factor, VEGF. VEGF is important for angiogenesis, and aplastin is the drugs that is often used to blocks the effect of VEGF. So if you don't have VEGF, you don't have angiogenesis. If you don't have any angiogenesis, the tumor can't get big, for example. That's another approach. People have tried anti-CSF1 in terms of angiogenesis. That's what my former mentor's working on. And then another characteristic of cancer cells that they have this ability to replicate to this passes the Hayflick limit. So they become immortal, if you will. So oftentimes this is refers to the upregulations of the enzyme called telomerase. It's very common in many cancer cells. Okay. And this is highlight some of those examples, for example, in terms of cell sufficiency, activations of the HRAS oncogene. That's they use that as an example in the Weinberg and Hanahan review, but it doesn't have to be this, right? It's just an example. But I would say an example is that they make their own growth factor. This is, however, an important thing, which is the word oncogene. Okay, onco for cancer and gene. So oftentimes in many cancer cell, there's an increase or upregulations of an oncogene. Okay, so increased expression. Yeah, so that's what oncogene is. It's a gene that is involved in oncogenesis or cancer, if you will. On the other hand, there is a such thing as known as a tumor suppressor. So tumor suppressor or normal function is to suppress, right? To suppress means to prevent cancer. And so in cancer, cancer cells usually have a mutation in a tumor suppressor, all right? RB, retinoblastoma, is one of these type of tumor suppressor. It was originally identified in children who has a mutation in RB, and they will have a retinoblastoma, which is cancer in the back of the eye. And usually if you shine a light, like a flashlight, the child's retina will reflect back, and it will shine. Typically, it's known as cat eye. Okay, so if you see cat eyes in children, that means they have a tumor, retinoblastoma, and what causes the retinoblastoma? It is a it's a mutation in an RB. Okay, the protein is called RB, retinoblastoma, and it's a tumor suppressor, right? So in cancer, mutation, mutation, and that means no production of a tumor suppressor. Whereas in the cancer cell, they upregulate or they increase expressions of an oncogene, right? So when I say, oh, a cancer cell, boom, you think of oncogene or a mutation in a tumor suppressor. Joseph, you have a question?
Yeah, so you're
Joseph?
saying the suppressor stops the tumor from. Can you hear me,
Yeah,
Professor?
yeah, I can hear you.
Yeah. So you're saying that the tumor suppressor stops the spread of the tumor for certain parts of the body?
Yeah, so that's its normal function. Its normal function is to stop cells from dividing. But if you have a mutation in a tumor suppressor, then you get cancer. You see that? Does that make sense? It's
So the
almost like
cancer
we was
caused
moving.
a little bit.
Huh? I'm sorry, repeat.
It makes a little bit of sense. Makes a little bit of sense.
Okay, yeah. So usually a mutation in a tumor suppressor will lead to cancer. Now, all right. So here's the normal functions of RB. RB, in a way, is kind of like P53. It's also a tumor suppressor, and that it does affect the cell cycle, right? And um, normally, what RB does, okay, its normal function people know now is like, oh, it stopped the cell from cycling. And in other words, what it's going to do is that, particularly if you have DNA damage, okay. So whenever you have DNA damage, right? So let's say here's a cell, right? Here's a cell, here's the DNA, okay? You have DNA damage. Let's say UV light. The normal, if there's a damaged DNA, the cell was trying to fix it, okay? Will fix it. So they do upregulation of a number of mechanism to fix the DNA damage. But right now, from the big scheme of thing, what you want to do is that you don't want this DNA damage to pass on to the next generation, right? Because then the next cells, the daughter cell, will inherit that mutation as well. So what happened is that now if we look at this is the cell, right? But the cell is involved in cell cycle, okay? And the cell cycle is that it spent a little bit of time in mitosis, but it spent most of its time here. It's called the G1, okay? And then it goes into the S phase. We talk about that, where it replicates the DNA. So you don't want to replicate it, mutated DNA, right? And so typically what RB and P53 does is that it stops the cell cycle here, okay? Is that, okay, do not goes into this S phase. Just stay here, okay? Allows the cells to fix the mutation and then goes on, right? Now, if your RB is mutated, then the cells just go through this. I'm summarizing it, but it's involving phosphorylation of transcriptional factors in essence. But think of it in picture. So then what happened is that the cells start to go through G1S transition, right? And then they started to divide. So in general, we can say, oh, there's a number of tumor suppressor, okay? So say loss, that means mutation. So mutation in a tumor suppressor, okay, will lead to cancer. So that's the insensitive to anti-growth factor. So in the context of RB, it's kind of interesting. Let's talk about P53. So I mentioned to you last week that P53 is the molecule of the year several years back, okay? In fact, now it says that in many tumors, we see this, there's a mutation in P53, okay? And P53 is because this is a size we run on an agarose gel P53. There's a mutation here. But what is the normal functions of P53? That's kind of like RB in the sense that here's a cell, here's a chromosome, here's a DNA damage, right? So one of the function, I'm going to write it down, the P53 is that if it sees the DNA damage, it will turn on the repair mechanism. Okay. And there are different kinds of mutation. It could be, say, mismatch reaction. Again, you know, ATGC, ATGC pairing for nucleotides. But if you have an A, you know, C, A happen to pair with C, or G pair with T, then that's not compatible. That's wrong, and it will probably cleave the nucleotide and prepare that. There are other kinds of mutation, like there's an insertion mutation. You insert another nucleotide in which the frame gets shift. There's a deletion mutation, all kinds of mutation. Okay, we're not going to talk about different kinds of mutation. Just say mutation. And so if it sees that, it trying to prepare that, okay, to repair that by fixing it. It is just fix it up, fine. Okay. Now, in order to fix it in a way with with RB in that sense is that it blocks the G1S transition, right? It makes sense. You don't want to go into the S from DNA, right? Now, let's say, for example, that there's too much DNA damage because when you have a lot of radiation, right, from nuclear plant, for example, then it's just so many mutation everywhere. The repair mechanism trying to fix it, but it can't because it's too much. In that case, then it would just say, okay, it's not worth to repair this, but rather let's commit suicide. And so that's the third function with P53 is that it does involve an apoptosis, right? So P53 triggers apoptosis. Right, so let me write this down, particularly if there are too much DNA damage. Okay. Okay, so that's it. So that's really the three functions of P53. Obviously, if you have a mutations in P53, it not blocks G1S because its normal function is to block G1S, and then you can the cell just cycle and cycle and cycle. Okay. So P53 is also another tumor suppressor, but there are many kinds of tumor suppressor. In general, if you have a mutation in tumor suppressor, you will get cancer, or you get an upregulations of an oncogene. Okay, an oncogene. RAS oncogene is one of them, but there are a numbers of oncogene that I myself have worked with. STAT1 might be considered an oncogene because if that thing is upregulated, you see a lot in cancer. Okay. Evading apoptosis, usually, things that are tell it to die, but it doesn't die, for example, that's called evading apoptosis. You can talk about this general survival of insulin-like growth factor, which is usually downstream of growth factor in the liver, but in general term, we call it evading apoptosis. For it to have limitless replication potential, usually upregulations of telomerase, that's very important. For it to have sustained angiogenesis, you have to have VEGF, or vascular endothelial growth factor. And remember this is a drug, aplastin is a drug to blocks the effect of VEGF. And in doing so, the big tumor, usually big tumor need a blood vessel to die. And if you block this, tumor can't get that big, okay? Tumor can't get that big. And for tissue, an invasion, usually it turns on stromolysin, okay, or digestive enzyme. In the general term is stromal. For stroma, lysin is protease that break. Stromal lysin. Okay. All right. So these are the 6 important factors that are associated in a lot of cancer. Grace?
I'm sorry, can you just repeat what the stromolysin is again?
Yeah, so the word stroma refers to the basement membrane. Okay, so here's a cancer. This cancer has a lot of cells. It's still considered benign if it has a stroma which is the basement membrane surrounded it. Okay, this basement membrane. Basement membrane is, in general, is known as the stroma. Okay, the stroma is not the cell, but is the component surrounding the cell. This is general term is known as stroma. And lysin is our enzyme that involving lysis, which is mean to break, right? And so the tumors put out stromolysin are enzyme, if you will, and they'll degrade the matrix. Okay, the stroma. They destroy this, so tumor cells now can go out. This is known as metastasis. Okay, so metastasis to metastasize mean to move right onto another one. So cancer cells, particularly the malignant type, metastasize. I mean, they're going to another site, distant site. And three important metastatic sites are, one more time, the lungs, the liver, and the bone marrow. So a lot of breast cancer, you get cells, usually stage 4 that has been metastasized. It goes to the lung because it's small blood vessel there. It goes to the liver, and it goes to the bone marrow. All right.
Thank
Does
you.
that answer your question? Okay,
Yeah.
you're welcome.
Thank you.
Good. So important to note that in general terms that cause of cancer are usually upregulations of oncogene. We call this upregulation. Now, what happened is that the normal genes location, right? So I don't know if you guys take any molecular biology or anything like that. Just a brief course. And that is, let's say here's a DNA, the chromosome, right? So when they say pro-oncogene, that means that in this chromosome, there is a pro-oncogene there. It's not quite, if it's active, upregulation of the then it become an oncogene, which is called cancer. But a pro-oncogene is not quite yet. It just sits in there. It's part of the chromosome. But every gene usually have what we consider a promoter, okay? And the promoter is what drives the expressions of the gene, right? So in other words, I don't know if you guys know transcription, RNA polymerase get in there, binds to the promoter, and it drives the expression of this. And this promoter is regulated by a number of operators here. It could be, like transcriptional factor, whatever it is that regulate this promoter. So upstream of the gene, there's a promoter region. A promoter is what drives the expression of the gene. It's a pro-oncogene. It just sits there. It doesn't express. If it doesn't express, it's not cancer. If it's expressed, it becomes cancerous. And so what happened is that, and we see this a lot, right? You can have either a virus that inserted into this upstream region, okay? And it has its own promoter, and doing so, the viral promoter that get inserted into the chromosome now activate this oncogene, and now you have cancer, right? So there are murine leukemia virus, for example, okay, MLV for murine leukemia virus, okay, is one example of a virus that will insert itself near the oncogene, right? And then by its presence, it changes the normal promoter. The normal promoter is probably not working, but having a virus sits to it, now it can turn it on. That's one, okay? Another type is that sometimes we see this happen a lot, okay? In which chromosome, usually chromosome are or number based on the size. So in other words, the biggest chromosome is chromosome number 1. Okay. And then you have 2 copies of chromosome number 1. Okay. Number 1. And the smallest chromosome is the Y chromosome. The smallest sex chromosome is the X, and then but the Y is even smaller. And let's say here's another chromosome, chromosome number 9. So let's say this is chromosome 9, and this is chromosome number 20. I know it doesn't work too well. Okay, so let's say that this is chromosome number 9, and this is chromosome number 20. And so what happened is that there is switchings of DNA, okay, switchings of the DNA, so that now part of the DNA of number 9, number 20, it goes into number 9, and then part of chromosome number 9 goes into the 20, okay? So this is the general term is known as translocation, translocation. Right. So translocation is that you just basically chromosome, part of the chromosome, part of the DNA in one chromosome is hop onto the other. The translocations of 9 to 22 or 22 to 9 is known as the Philadelphia chromosome, which is involved in CML for chronic myelogenous leukemia. And we'll talk about this when we talk about blood disorder. But it's very common translocation. And essentially what happened is that there is a gene that usually a tyrosine kinase that sits in the area. It's inactive, but after translocation, it becomes active. So then it's acting as a proto-oncogene as well. So upregulations of tyrosine kinase, and this is known as the JAK kinase. So translocation is another method, if you will, of how you might activate the proto-oncogene. Tumor suppressor, we talk about RB or P53. P53 is also a tumor suppressor, but it does more than just regulating apoptosis. It stops the cell cycle. It's upregulating the DNA repair mechanism in general. Okay, so carcinogenesis is you define them as stages or steps involved in the development of cancer. It's certainly a multi-step process, not just one thing, but many things. And by the way, the Weinberg and Hanahan review, and it tells you that there are those 6 features. Usually there's multiple things that is happening, right? You can have cells that is upregulating stromolysin at the same time evading apoptosis at the same time. So there's multiple things that is happening at the same time. Upregulation of telomerase, but it doesn't have to mean that every cancer have to have all 6 of those factors. Just 6 of those factors is very common. That multiple factors can be seen in one type of cancer, okay, but it doesn't have to be all 6. So most cancer-related mutations occur somatic. Okay, somatic cells meaning not germ cells, and so malignancy disease developed through pre-malignant state. Okay. And that pre-malignant stage is usually you set it up. We can say that benign, if you will, benign state. So a typical carcinogenesis, I kind of told you already. This is also good to know. For example, you might have a lesion that become hypoplastic. Okay. which means more cell number which become dysplastic. Okay, dysplastic, which is darker nucleus staining. The cells sort of like pile on top of each other. They kind of lose contact inhibition. We call that. They become carcinoma in situ. I would say that with the carcinoma in situ, the stroma is present. So it's still considered as very benign, right? Benign. And then it becomes metastatic cancer. Metastatic cancer. Okay. So this is typically, my research on colorectal cancer, and this is what the events that we see. You can define it as carcinogenesis. They become hypoplastic cell, dysplastic cell, carcinoma in situ, and metastatic cancer. Now, within the hypoplastic cell here, right, there are many cells, but they're not all the same, by the way. Okay, in fact, a lot of these cells are different from each other. Okay, different from each other. They're not the same, and so given whatever the environmental pressure is put on there are some cells that then grow, that will grow, selected to grow, for example. Okay, we have the selective advantage, right? And so then you can say, oh, it seems like, one of the feature of cancer is that is very, very similar to what Darwinism is at. In Darwin, theories of evolution is that there is, first of all, you have changes within the population, and the environmental pressure select for the species that is give it the advantage of surviving. So this is very similar. You have the diversity there first, right? This is the diversity, and the environmental pressure then is selecting out those that do not survive in this environment will not do, but those that can survive will proliferate. And here you can see that the yellow one proliferate or the orange ones proliferate. Okay? And so one of the feature of this malignancy is that there is heterogeneity. Heterogeneity means there's different population. If it's just one population, then it is known as homogeneity, but it's not. It is heterogeneity, and that is not only you have one, but you can have several that's selected to divide. And those cells that do divide, typically, in fact, you can look at the type of tumor, okay? And cells that are divide usually we can see them during the process of mitosis. That might be at the middle of the metaphase, for example. Okay. And so this tumor has 3 cells that we know that it's dividing because it's in the metaphase. Will divide much faster than if, a tumor that you see that, but you don't see very many mitosis. So depending on the number of mitotic cells, okay? So in this case, you see that there are 3 mitotic cells, right? Then that means that this one will have faster growth than this one that has no mitotic cell or cell that is dividing. Okay? So the growth rate of a neoplasm depends on the number of dividing cells. Another word for dividing cell is known as mitotic cell. So, right? So in the future, if I say, this tumor you can see a lot of cells in metaphase, then that means that that it's going to be rapidly, it's going to divide rapidly. as opposed to a tumor that you do not see any mitotic cells at all, then they're kind of like slow
Here's what
growing.
I found.
All right. So when you're looking at the lesion, you can look at the cells and finding how close is it to the parenchymal, how close is it to the original cell. So, all right, what I mean by that is that, let's say you have a lesions in the liver, and you look at the cell and say, oh, does it look like hepatocyte, which is in the liver, or is it undifferentiated? Okay, lesser degree, if you of differentiation, if you will. So in other words, either greater differentiations or lesser differentiation. And I would say this: in the tumor, right, or a lesion, let's say this, differentiated cells in the tumor is easier to treat than undifferentiated cell. Okay. If they're undifferentiated, then they kind of like go back to their original state. But differentiated cells tend to be easier to treat than undifferentiated cell. Okay? I can tell you that. Here's an example. One of the problems of treating breast cancer now is that there are breast cancer cells, we call it triple negative. Okay, triple negative red blood cell, triple negative breast cancer cell. And triple mean because it has, they don't express estrogen receptor, progesterone receptor, or human epidermal HER, okay, or receptor. HER is stand for human epidermal growth factor. All right, so triple negative meaning that they do not express three things: estrogen receptor, progesterone receptor, or HER. Okay? HER stands for human epidermal growth factor. Certainly, cancer cell, breast cancer, require EGF. So typically we call this EGF, epidermal growth factor, for it to grow, right? And so somehow the cancer cell now doesn't need epidermal growth factor; it just grows. They could produce more of it, or they might constitutively active the receptor. That's another component of you can think of. In other words, let's say here's a tumor cells, and here is the EGF, epidermal growth factor receptor, right? And usually, if there is an epidermal growth factor binds to this, then you get an amplification signal, usually phosphorylation that is occurring like this, right? But somehow it's possible then that the tumor doesn't need epidermal growth factor. In other words, there is a mutation in the receptor, or somehow this receptor can activate, can undergo this pathway in the absence of epidermal growth factor. Okay? There's a mutation. So we call that constitutively active receptor. Constitutively active receptor. So in other words, the receptor is activated. It's always on. It doesn't need the epidermal growth factor to turn it on. It's always on. And so this cancer then, may have this, or sometimes the cancer do not have that. Right? The cancer do not have it, or do not have this, or do not have estrogen receptor or progesterone receptor. If they don't, then we can say, oh, they're not differentiated, because one of the characteristics of breast cancer, if they differentiated, they would produce estrogen receptor. If they do, then it's easy to treat because you can give it tamoxifen. Okay, tamoxifen is the estrogen, is the estrogen analog. Okay, so tamoxifen is an estrogen analog, and so if the cell produces estrogen, so we call it ER positive, right? You can give the patient tamoxifen and they're responding to it. They're like, okay, if the cancer cell require estrogen to survive, right? If it's require estrogen in the estrogen receptor, then you treat it with tamoxifen. But if the cancer do not express this estrogen receptor, right? How do you give it a tamoxifen? Won't, you know, it doesn't express, right? And tamoxifen won't do anything to it. All right, take home message: differentiated cells are easier to treat than undifferentiated cells, right? So when you look at a lesion and say, oh, it's expressed this, it's this, boom, that's good, because it means it's treatable. If it doesn't, then it's not treatable. Neoplasm, certainly every cancer cells require nourishment. Malignancy, so associating malignancy means metastatic, um, that you have to upregulate the stromal. That's usually invasion metastasis, a hallmark of malignancy. And another important thing is that there are immune cells that seize these cells, right? They're baying, right? They're surveilling, uh, so they make sure that they keep the cell from uncontrollably. And typically, usually, if you have that problem of the suppressor T suppressor cells not working, or T regulatory is not working, then you get cancer, if you will. Okay? But in general, immunosurveillance is around to fight against that cancer. But usually, if that goes away, then you get full blown. Um, the couple of years ago, um, there was a Nobel Prize was awarded to um, um, what's his name? Um, but he's working on I can't think of his name. Is Alistair? Yeah, Alistair, James Alistair, who works on T cells that keep these. The T cell has a ways to put a break so that it doesn't kill your cancer cell, for example, or doesn't kill your normal cell. And so what James Allister did was that he tricked the system, it's involving CTL receptor, so that now it remove that break, and so the T cell, cytotoxic T cell, can go and kill the cancer cell. So this is the way of using your immune system to kill the cancer cell. In that case, it's a cytotoxic T cells. Okay. Jim Alistair work. Okay. Carcinoma. When you say carcinoma, particularly in situ, is still confined, right? It's considered benign. I would say then, so this is probably the stroma here. This is stroma here. And when these cells break away from that, because they're upregulating enzymes, then they become the metastatic cancer. This is kind of, it's gonna be there. It's pre-malignant right here. So that's going to break away and it's invade particular here, I think this is, if this is cancer down here in the stroma, that's metastatic right there. Okay, that's metastatic. And you see here's the basement membrane. So the basement membrane is a stroma. Okay, it's a stroma. All right. Normal epithelial, so basically similar carcinogenesis, dysplasia, carcinoma in situ, and you can see how the basement membrane is intact. It is being destroyed, right? Invading, right? And invading can either goes into the blood or can go into the lymphatic. Okay. And whether they go into the lymphatic and in the blood, it depends on what kinds of tumor. In the breast cancer, usually they're invaded through the lymphatic more, if you will. But they can go into the blood, but they tend to go into the lymphatic first. And so when a female feel lumpectomy, feel a lump, and if it's suspicious, you can remove it, called lumpectomy. But when you see a lesion, particularly if it's if the skin is attached to it and it's very wrinkled, then that's not a very good sign. It's called, we call it stell-like, okay? And you can see this in MRI, for example. And to note whether it has been metastasized or not, what you have to do is you have to remove the lymph node and see whether there's cancer cells in the lymph node. Okay, and so that's part of the staging. So I'm going to talk to you to differentiate between grading, which is an anatomical exercise, versus staging, which is the clinical definition. And staging depends on TMN. T for tumor size, N for node, whether it's node positive, and M for metastatic site. So we call it TNM. Okay, TNM. All right, doubling time. So essentially, here is this is normal person and 20 years old, smoker a pack of cigarettes every day, and you can see that normal cell over time, years, now have reaches, you only have one malignant cell that then divide, even though it looks like it comes from one, but it's changing constantly here. Okay, this is heterogeneity. It's not homogeneity. It could be one from here, or it could be other cells that give rise to another population. But note that by the time you're getting a tumor size, it's multiple clonals, if you will, right? Or they're constantly changing here. So they're no longer homogeneity, but heterogeneity. And once it reaches the 1 centimeter size, okay, 1 centimeter size, usually it's the finger length. Yeah. And from that so from one cells to this one centimeter size, it could take years, 20 years, 30 years, 40 years, or 50 years. But once you have this one tumor size, 1 centimeter size, to reach this to metastatic site, it's in a few years, something. You see how the graph of this thing is like kind of like exponentially, if you will, right? So it could take years, but once you reaches these 1 centimeter site, and it just basically boom, right here, exponential phase growth. And so from here, from 1 centimeter size to metastatic site, it just takes months. You don't take years anymore. And that's the take-home message. So essentially here, this is considered mitotic cells. So in this lesion, you have 1 mitotic cells, whereas this one you have 4 mitotic cells. Certainly, the lesion that have 4 mitotic cells will have faster growth rate than the one, right? So we call this is one dividing cell out of 5, so 20% growth factor. Well, this is 4 out of 5 or 80% growth factor. So growth factor, growth fraction. So usually this is low growth fraction, and this is high growth fraction. Okay, fraction is a fractions of the cell that is mitotic. So usually this one have faster growth rate than this. It makes sense. But this is observable. You can see this. All right, differentiated versus undifferentiated. So what you can see here, this is the normal hepatocyte within the liver, right? They forms these kind of like leaflet where here's a sinusoid, right? They're differentiated kind of large cells. You can see it. So in other words, they're differentiated. Right? On the other hand, if you look at the lesion called hepatocellular carcinoma, I mean now that is very aggressive, metastatic site, if you will, potentially going to the metastatic site. And you look at these cells, it's kind of hard to define what they are, right? So they're undifferentiated. What we do know is that there are some cells under mitosis, so then that they have a lot of mitotic cell, that means they're fast growing, right? And this is really hard to treat. One of the issue with when you see a lesions in the liver, it's hard to determine whether, or you always keep in your mind whether, is this the tumor that starts in the liver, or is this a metastatic site that the tumor starts from somewhere else and it comes and stays into the liver? That's something you always have to keep in mind, is that when you see a lesion, you got to make sure that it is, liver or not metastatic cell that goes to the liver. So keep in mind that differentiated cells is easier to treat than undifferentiated cells. What are some of the ways in which how the cells might move away from the initial site? Okay, there are 3. You can see here is by seeding. So this is the ovarian cancer that have just kind of like, crawling to the nearby organ. We call that seeding. Or it can basically move through the distant site, through the lymphatic or through the blood. If it's more carcinoma, I would say that it is through the lymphatic. Okay. If it's sarcoma, that's like osteosarcoma, then that travel via the blood. Okay, so keep that in mind. So sarcoma or mesenchymal origin, which is mesodermal, as opposed to carcinoma, which is more epithelial. Like breast, for example, is epithelial, and they spread via lymphatic. They do some to the blood, but mainly is through the lymphatic. Grace, you have a question?
Yeah, so they say a lot of times when women get breast cancer that they have a higher risk of getting ovarian cancer. So is this because of like seeding or any of these?
No. So I don't know if I talked to you about Mary Claire King, who has the same mentor as I do, Mary Claire King, and she was working on family with breast cancer. Right. So in other words, what she did is she looked at family who either aunt has breast cancer, the grandmother has it, that potentially the daughter might have it, and so forth. But through many generations, if you get the DNA, you can map it and see, where did these people have a mutation? Obviously, it's hard work because there's a lot of changes in the DNA, and how do you know what is mutated and what is not, right? And so that's it's hard work to trying to sort through what does these women who have cancer that the normal people do not have. What is it? How is it related to DNA? So in fact, it was discovered that actually she identified them. They call them BRCA, BRCA1, and now we know there's another one called BRCA2. And if you have a mutation in these two, then you have your increased risk of developing breast cancer as well as ovarian cancer. And so, no, we're talking about the tumor in the breast and the tumor in the ovary that become cancerous, not that the breast cancer cell has spread it to the ovary. No, we're not talking about that. That's spreading. And I don't think that you were talking to that. But the reason I'm saying that breast and cancer often go hand in hand is because they have a mutation in these BRCA1 and BRCA2. And Angelina Jolie, for example, knows that she has a mutation in BRCA1, right? And so that means that she increased risk of developing breast cancer and ovarian cancer. So what she decided to remove her breast, mastectomy, and ovaries, so ovarectomize her ovary as well. So she can't have kids, so she adopt kids. All right, fine, adopted kids. Did they have any children with Brett? I don't know. Okay, does that answer your question?
Yes, that definitely does. Thank you.
Okay, you're welcome. So different ways that cancer cell might spread to distant organs. And again, what are the three common site, distant site? The lung, the bone, and the liver. And if the tumor is derived from epithelial, like carcinoma, it usually arrive at the metastatic site through the lymphatics. If it's more like mesenchymal origin, like sarcoma, then it via the blood. Okay. Breast cancer and colorectal cancer, these are all carcinoma, epithelial. Then the invasive is to the lymphatics. And here, just basically looking at the lymphatic vessel, kind of very similar to vein, because the wall is very thin, right? There might be some here, and you can see how cancer cells are finding in there. And cancer cell, oftentimes, remember, the nucleus is going to be darker staining, right? So we also say is heterochromatic, hyper, not hetero, hyperchromatic. That means more staining, darker staining. All right. Now, the three most common metastatic sites I talked about already: lungs, liver, and bone marrow, because the lungs are small sinusoids. That's where cancer gets stuck. Cancer cell gets stuck. Liver, you can see here metastatic cells, and it's hard to determine whether is this really metastatic cells, like from the breast that goes to the liver, or is it maybe it is the liver cells that become cancerous like this? So how do you know? How do you know which is which? Well, if these right here express this estrogen receptor, for example, right, and they're more epithelial origin, then we think it is breast, right? But it is because the liver is not by any means epithelial origin, and so you won't see any cells that expresses epichem, for example. Okay, epichem is one of those self-adhesion molecule that only finds in epithelial cells. So that's how, an example, you might differentiate between metastatic cell versus the liver produces own that become cancers. All right, so why don't we take I only have a couple more slides, and then we'll take a 5-minute break, and we'll come back and finish this, and then I'm going to talk about infectious disease. If I don't finish that, I'm going to finish it next week. But at the same time, this is your tough week. I'm going to post a recording on fluid dynamic, and that is because next week we have to finish those infectious disease and fluid dynamic, because on the 7th, on the 24th, you have your second exam. Okay, so 2 weeks from today is your second exam on those little bit on immune disorder, but typically cancer, infectious disease, and fluid dynamic. Are there any questions? Okay, let's stop here. I need to take something to drink. 5-minute break, guys.